Okay, so once you're in Google Classroom, you'll see that I've posted a new assignment in the stream. Okay, if you click on that or if you click on classwork, either way, okay, it'll take you here. Okay, the properties of compounds lab. This first file is just the PDF. It's the lab sheet. All right, I'm going to be working from that as I explain some things to you guys. Okay, this is the lab report file here. It doesn't say student Coderre for you. It says your name. Okay, and then U1 properties of compounds lab report. That is the report you will be submitting to me electronically through Google Classroom. That file and only that file. Okay, don't make another one at home in another program. I won't mark it. Okay, it needs to be this one because this one I built for you. It's going to sound creepy, but I can see everything you do on that file. Okay, that's the whole purpose of it. Okay, I need to know, if you make some mistakes, I need to know why. I usually go back in your editing history and kind of see what kind of things may have caused you to make mistakes and things like that. I can only do that through that file. So I need you to use, submit, fill out that file. Everybody clear on that one? Okay, so when you open it up, okay, it looks like this. It's kind of like the order of the lab stuff we went over on Friday. Okay, we said these are the things that you're going to include in a lab report. Okay, so I've built the template for you. All you have to do is fill it in. All right, so I'm going to save you some work here. Okay, all that you guys have to do is fill everything in as we go along. All right, so as I go through the lab sheet, I will give you some pretty big tips and hints for what kinds of things I'm looking for, similar to what we did on Friday, okay, with the example one that we went over. Okay, is everybody to this point here got this opened up and ready to go? Okay, so on Friday we talked about those lab report expectations and we went over the properties of different types of compounds. Okay, and I said in that lesson a lot of things were going to be important for assembling this lab report here. All right, because on Thursday we will be in the the biology lab. Okay, and you will have about a dozen unknown samples on the front table. Okay, they'll be labeled A through K or whatever it is. What you will have to do is run the tests that we talked about on Friday, okay, an observable properties test, okay, you'll run a solubility test, an aqueous conductivity test, pH test, all of those things, okay, in order to get as many of the physical properties of that material as possible and then use that to identify what material that actually is. All right, so we're going to kind of build to that right now, but we're going to start off with the problem. Okay, we're going to move to the hypothesis, what that's going to look like, the variables, all of that kind of stuff. Okay, so follow along. You'll want to type some things in as we go here, because like I say, this first lab, I pretty much hold your hand, okay, through the whole thing. Okay, so you should do pretty well, okay, on those parts. All right, so for the problem, remember we said you need to restate this in your own words in the form of a question. All right, so the one I have on the lab sheet is, given several unknown samples, how can we classify them as ionic, molecular, acids, or bases in order to find their identity? Okay, I would say there's probably a way to simplify that. I would say that your problem needs to be a question about using physical properties to identify compounds. Okay, how are you going to do that? Something along those lines is what you need to write as the problem. Okay, a question that says how are you going to do that? Okay, the whole point of the lab is to identify unknown compounds, based on just their physical properties. Okay, for this lab, okay, the way this lab is going to work, and this is how I'm going to explain the variables part to you. Okay, the way that this lab is going to work is you will get, and in fact, all right, so as I was saying here, the way this is going to work is you are going to get a depression plate. Okay, that's what this is. It's not sad. Okay, it's just got little pits or depressions in it. Okay? That is where your unknown samples are going to go. All right, so how big are the sample sizes going to be? Pretty small. Okay, we don't need them to be very big. Physical properties don't require us to have a whole kilogram of something. Okay, we just need to be able to see it. Okay, see if it has an odor. Okay, look at it, if it's got crystals or not. Test whether it can dissolve in water, which we can do in here. Okay, we can test the conductivity. Okay, that's what this device is for. All right, and like I said, it has a scale of 1 to 10 in terms of the conductivity. Okay, and all you do is press the button. Okay, the on light will come on. which makes these ones vastly superior to the ones we had before, which didn't have an on light. So if the battery was dead, you put it in, it read everything as 0. Okay, and you just assume nothing conducted because you didn't know any different. These work better, right? So when you put them in something that does conduct electricity, then of course the rating will go up, all right? For ionic compounds, basically anything like 7 or above is ionic, okay? And anything that's like 4 or below is going to be likely to be molecular, okay? They may have some small amount of conductivity, but okay, it's going to be minimal. Okay? Most of your true molecular compounds, only the on light will come on. Okay, everybody with me there? All right, so that's what we're looking at for the conductivity tester. Okay, and then we've got our Hydrion paper or our pH paper. Okay, it comes in these little orange strips. Okay, one of these strips should be able to test 2 unknown compounds. Just rip it in half, dip it in the compound, and then compare it to the color chart. Okay, that's on the container, right? Pretty straightforward there. Okay, just match it up with whatever the color is, and then write down that pH. Okay, so that's the equipment you're going to be using. Okay, so we're going to run those same tests. Okay, the same set of tests we talked about on Friday. We're going to look at observable properties, then run a solubility test, then run a conductivity test, and then run a pH test. Okay, those are the ones we're going to do, and we're going to do that for every single compound. Okay? Every single compound is going to use those same tests. So the only thing that's different every time we run the tests is the substance that we're testing, right? That makes the substances we're testing which variable? It makes them the manipulated variable. You're welcome. I just laid that out for you. Okay, but okay, the unknown substances in this lab are our manipulated variable because we're testing each and every one in the same way. Okay? We're running the same battery of tests on every single one of them. Now, for your explanation, obviously, you're going to need a little more than we're testing them all the same way. Okay, we're looking for an explanation here that says, how are you manipulating them? Okay, they're going to be different. It's a different one every time. Okay, why is that? Okay, make it the manipulated variable. Okay, what's the effect that's going to have? Okay, and you might not know all that right now. Okay, but those are the kind of things that need to be in there for right now. Okay, in terms of responding variable or variables in this case, each time I test a new material, what should happen? Okay, actually, I'm hoping to not have too many reactions, but I should get different results, right? I mean, everything should look at least somewhat different. I mean, I might have a few white crystalline solids, but I mean, they're all going to be somewhat different, okay? Not all of them are going to conduct electricity. Not all of them are going to dissolve in water. They're not all going to have the same pH. Every time I test one of those new materials, I should get a different set of physical properties. Okay, so that makes physical properties which variable? Makes it the responding variable. Okay, and in the case of this one, for your explanation, you want to talk about how it's the physical properties in general that will be different. Okay, it's possible for some of them to have similarities, like all the ionic ones are going to dissolve in water and conduct electricity, but there's going to be their set of properties will be different. Okay. Now, controlled variables. We need to list 3 and explain each one separately. Now, I've kind of made a big deal so far here out of what we're doing to each compound. Okay, remember our controlled variables are the things we keep the same throughout the entire experiment or for each subject. Okay, in this case, each subject is a different unknown material. Are we testing them all the same? Okay, is that important? It would seem to me that it would be important to test them all in the same way. If I don't, I won't get the same data set and it'll be hard to compare them. Okay, so in this way, the battery of tests or the set of tests we are running must be a controlled variable so that we get similar data sets for each unknown material, and we can make a fair comparison. Controlled variables are all about keeping an experiment fair. Okay, that is being able to fairly compare everything you do. All right, so that's one. I need you guys to come up with two more, not right now, but at some point during class, you need to come up with two more controlled variables. I want you to think about things that could affect our tests. Okay, they don't have to be things we're actively keeping the same, but as long as they are staying the same throughout the experiment, okay, that'll be something that'll be a controlled variable. All right, I want you to think a lot about what kind of things might affect solubility, for example. Okay, if there's anything that can affect how much of something dissolves, that should probably stay the same for all of the materials. Okay, so a lot of controlled variables will be environmental. Okay, they're not things we're actively controlling, but they're being passively controlled. They're not changing during the experiment. Okay, they can also be methodical. And that means they're part of what you do in the experiment, how you perform the experiment. Those are methodical controlled variables, and then there are environmental controlled variables. And you can think about what are some things that we're doing the same. The one I gave you about the tests is a methodical controlled variable. Okay? It has to do with the method or the experiment. Okay? I would say you should be able to come up with two more that are probably environmental. Okay? All right, questions so far? Okay. What three words did we say our hypothesis needed to contain on Friday? If and then. Okay, our hypothesis needs to contain the words if and then. All right, so the premise that we're working on is what goes in the if part. Okay, so the premise we're working on here is that all of those compounds that we have different physical properties. Is that a fair premise to work on? If they don't, it'll be impossible to separate them, right? If they're all white crystalline solids that dissolve in water and conduct electricity and have identical pHs, they're probably all the same thing. Okay? So the premise we're working on here is that all matter has unique physical properties. Okay? Does anyone remember why that is? We talked about it the first day. It had something to do with the atoms. Right. All atoms, all compounds are made up of different materials, and all materials have different physical properties. So, okay, because of their atomic structure, they're going to exhibit different physical properties. Okay, so this is the premise we're working on. That should be part of or all of the if part of your hypothesis. If that's true, okay, and what did we talk about? Sorry, I'll let you type for a second here. I'll pause. Okay. Okay, the and part of our experiment, or sorry, the and part of our hypothesis, sorry, describes the experiment. Okay, what are we doing in this experiment? Okay, we're testing some stuff about compounds. What are we trying to find out about them? Their physical properties. Okay, so the and part is just a brief description of what you're going to do. Okay, a very general description. Okay, in our experiment here, okay, basically we're going to run tests to discover the basic physical properties of these unknown compounds. Okay. Okay, so the and part is just like we're not going into detail, right? That's what the procedure is for. We'll give people detailed instructions then. This is just a brief description of what we're going to do. Okay, and that's pretty brief. the then part of our hypothesis needs to tell people what our expected results will be, what we are going to be able to do, okay? Basically the end product of this experiment, okay? So I would say in this, we should probably tell them the kinds of things or the kinds of patterns we're going to see. Maybe we can talk about how ionic compounds will do something that molecular compounds won't, okay? Or that each compound should have a unique set of data, okay? And that will allow us to do what? Identify them, okay? That's the whole purpose here. We're trying to identify these compounds. That's the then part, okay? We're going to be able to do that because of this, okay? So in the then part, okay, what will we find out and why? Okay, so what we're going to find out here is the identities of the unknown compounds because, and then you talk a little bit about, that ionic compounds do this, molecular compounds do that, acids do this, bases do that. Okay, a little blurb in there about some of the specific examples of observations you'll get. All right, so we've got the then part there. Okay, we're just going to go back to this. Now, below that and in your package there, okay, so the file that I gave you, you've got your materials. You don't need to change those at all, okay? In these materials are obviously the depression plate, the pH paper, the conductivity tester, okay, things like that. But also all of the unknowns are here, okay? I have listed them as materials. You don't know which one will be which, but they're all there. Okay, so we know that one of the unknowns is ammonium nitrate. One of them is paraffin, which is candle wax. Okay, one is hydrogen chloride or hydrochloric acid. Okay, one is cobalt 2 chloride. Okay, one is distilled water, and we'll also be using distilled water for the solubility test as well. So it's a material on two fronts. Okay, copper 2 sulfate is one of our unknowns. Sodium chloride, which is table salt, is one of our unknowns. Sucrose, sugar. Okay, one of our unknowns. Nickel 2 chloride, one of our unknowns. Ethanol, okay, which is alcohol. Sodium hydroxide, that's the stuff that was in oven cleaner, okay, and it's one of our unknowns. And then obviously we have water again. It's distilled water, but I've got it on there twice because we're using it in two different forms. We're using it as an unknown, but we're also using it as a solvent for our solubility test. Okay. The other things we have in there, scupula, thermometer, okay, conductivity apparatus, pH strips. I don't have depression plate in there, but it should be. Okay, those are our materials. Okay. So the next thing that you have to do, and I'm going to let you work on this with some other people, is you need to design the procedure for this lab. Right? We talked about the things that we're going to want to do, okay, and that is these. Okay, we wanted to run an observable properties test. Okay, and in that observable properties test, we wanted to look for things like color, state of matter, whether it was a solid, liquid, or a gas, the nature of the matter, was it crystalline, cloudy, waxy, soft, hard, okay, whatever. Okay, odor test, which we would waft. Now, here's the problem with the wafting test. I've got all the unknown samples on this little depression plate. How good is a wafting test going to do? Not very good from this. Could I waft the original samples? That's what you would want to do. Okay, there's only really one of them where smell is going to be important. Okay, but you still need to waft them all to record something. But one will have a very strong distinctive odor. A second one may have a slight odor. Okay, but it probably won't be all that helpful. One has an obvious odor that will be very important in helping you identify it. Okay, but I would waft the original samples because they're far apart. Okay. Then we said we would want to run a solubility test. Okay, so we need to look for things like, does it dissolve? Is there a temperature change? So we would have the thermometer in the sample as we add the water. Okay, to make sure that we catch any temperature changes. Okay, any color changes, any new odors that might get produced. Okay, conductivity test. Once we've done the solubility tests, okay, then we just take our conductivity apparatus and we stick it in each of the depressions. Okay, test it. What should I do in between? Yeah, with tap or distilled water. Distilled water. Rinse it with tap water. You'll put ions on the leads and you'll get everything conducting electricity. So you want to rinse that with the distilled water, which will be in little squeeze bottles. Okay. Once you've done that, okay, then you can run your pH test after that. Okay, pH strip in each one. Okay, testing those. Now, those are the tests we want to run, and we talked about those the other day. I'll put this back up later. Okay, but when we do our procedure, we need to include everything. Okay, if you recall, I said we want to write this procedure as though the person who's writing the test is like the dumbest person you've ever met. Okay, you need those instructions to be so explicit that there's no way they can screw it up. They have to be idiot-proof. Okay? I know that's hard because an idiot will drag you down to his level and beat you with experience. You have to remember, okay, that you have to make these instructions so explicit that they don't think. Okay, they don't think for themselves, they just follow the instructions. So that would need to start out with things like, upon entering the lab, grab a set of safety goggles and place them over your eyes. Okay, that's kind of important. Okay, don't say put them on. I've seen people do this. They think safety goggles go here. Last time I checked, they're not a forehead protector. Okay, they need to be here over your eyes. Okay, you need to be explicit. Okay, you can even say grab a lab apron. All right, you put on a lab apron and it'll give you some protection on the front of your clothes. Okay, in case you spill anything. Then you'd say, acquire the depression plate and conductivity tester, etc., from the front table or something like that. Tell them, go get your materials. Okay, and then I would say things like, if I want to use this depression plate and I've got all of my samples in it, what should I make sure I do to this plate? Okay, yeah, be sure not to mix them. And if you do, it's not a big deal. Just get a new sample. There's more than enough holes here. Or when you're done, just dump it all out and try that one again if you screw up. Okay, and that's a big thing. If you screw up, just do it over. Just do that one over. Don't try to rescue it. Don't try to save the materials. It's wrecked. Okay, if that happens, and it always does, just do that particular one or both of those ones again. Okay. But if I've got 12 different samples in this plate, what should I do to each depression? Write it down. Yeah, label it. I'll give you a Sharpie. Okay, you can write right on here with a Sharpie. This one's A, this one's B, this one's C. Okay, so that you can match them up with the unknowns that are on the table. Otherwise, you'll forget which one was that again. Then you'll be running up front. Oh, it looks like it was probably that one. Probably is not a word we like to use in science. Okay, definitely, we like that a lot better. All right, so label it. I'll give you a Sharpie so you can do that. Okay, again, that's something you'll probably want to have in your instructions. Be explicit. Label the depression plate. Okay, go up to the front, grab small samples. Now, how much sample should I put in here? Should I fill this thing all the way up? Not if I'm going to run a solubility test where I add water. Right? I would say probably the most you're going to want to fill this is about a third. Okay, even that's probably lots. Okay, so fill the depressions about a third of the way full with sample. Okay, then I would say you take it back to your table and you do which test? The observable properties test. Okay, now's the time to do the observable properties. Can't do it after you add water to everything. So now's the time. Okay, when you're telling them how, you don't just write down step 3, do the observable properties test. They're not going to know what that is. That's not an idiot-proof experiment. Okay, it needs to be explicit. Okay, performing the observable properties test means tell them you're looking for these things. Okay, and you can go back to this list here. Okay, in the observable properties test, we are looking for record, or you could say record observations of color, state of matter, okay, and solid, liquid, or gas. Nature of the matter, is it crystal, is it cloudy, is it waxy, is it soft, okay. Odor test, perform odor test on original samples by wafting, okay. Record any observations, etc. Okay, be explicit. Tell them here's what to do, here's what to record, so that they know, right? Because you can see in your copy of the lab report, you've got, okay, an observations chart here with lots of room for the observable properties test. Okay? We don't need a lot of room for these results. Solubility is basically yes, no, or slightly. Okay. Conductivity is a number between 1 and 10, and pH is a number between 1 and 14. We don't need a lot of room for those. There's lots more room for observable properties. For tomorrow, I would say you need a hand-drawn version of that chart to take into the lab, or sorry, for Thursday, to take into the lab, all right? Because you can't take the Chromebooks into the lab. They'll get wrecked in there, okay? So have that on paper, okay? Or at least a rough version of it on paper, and then you can transpose that later. Okay, and then once we have those observable properties, okay, then we'll perform the solubility test. So I would say that for your procedure here, you're probably going to have your, your first couple steps about what you do when you first get in there and where you get your materials, then how to perform the observable properties test. How many should you do at once? Yeah, probably one, okay? Do the observable properties test for sample A and record the following things. Then perform the solubility test on sample A, okay? Record your observations. Now, when you say record your observations, what are some signs that something's dissolving? Okay, there might be some bubbles, okay? Okay, if it's a liquid and a liquid, they're not separated, one's not floating on the other, okay? If it's crystalline, how do I know it's dissolving? It might disappear, okay? In this case, because we have such a small sample area, it probably won't all dissolve, but you could certainly see that the sample size shrinks, okay? The crystals maybe change shape, become more transparent, okay? Things like that are all things to tell the person to look for. Obviously you're also going to be recording the temperature, so you would record any temperature change that occurs as you add the water, okay? Things like that. Those are the things we want to tell them to observe and record, okay? Or tell ourselves to observe and record when we're doing this. Okay. After the solubility test, do I keep that solution I've made? Yes, I do, because the next two tests require it, okay? The next thing I'm doing is testing the conductivity of the solution I just made in the solubility test. So I've got this solution of A and water, okay? I press the button on the conductivity tester, I place it in the solution, and I record the number. That one's an easy one. You just tell them, place it in, push the button, record the number, okay? That's all they have to do for the conductivity test, right? After the conductivity test, then they do the pH test, right? Since you'll have, several steps here, probably like 7 or 8 steps on how to do all those things for one sample, what do you write after that? Exactly. Repeat steps 3 through 7 or whatever it is for all other samples. That saves you a lot of time because I've had people like copy and paste it 12 times. Okay, the report's like 15 pages long, and 8 or 9 of those pages are the same damn thing. Okay, you don't need to do that. Just repeat steps, whatever, for the rest of the samples. Should you tell them what to do after the experiment? What should you do after the experiment? Clean up. Okay, you always got to clean up. If you have your fun, then you got to clean up. So, okay, you tell them things like, take the unknown samples to the waste bucket. These don't go down the sink, right? Cobalt and hydrochloric acid and sodium hydroxide cannot go into the sewer system, okay? They're very bad. So they go into the waste bucket, which is in the fume hood. The waste bucket is, you'll know, it's the disgusting bucket in the fume hood, okay? It looks like, yeah, mad science gone awry. Okay, so I'll show you where it is on the day, but that's where all the stuff will go. And you'll rinse this off with your distilled water so you get all of it into the waste bucket. Okay, as long as you got most of it off, then you can go and finish cleaning it in your sink. Okay, so that's another part of the procedure. How do you clean up? Okay, you do this into the waste bucket, wash and dry everything, and return it to where you got it from. Okay, everybody clear on that? Okay, so that's going to take probably the bulk of your time today, okay, is getting that procedure done, but you can work on that with some other people, okay, in order to come up with some good ideas and good wording on that one. Okay, so once you have the procedure, like I say, Thursday we'll acquire all the data, and then once we have all the data, okay, then we'll be using the analysis chart here. All the analysis chart is you're telling me whether sample A was ionic or molecular, and what its identity was. So let's say that sample A was a white crystalline solid that dissolved easily in water, it conducted electricity, and had a pH of 6. Okay, I would say that's probably salt, probably sodium chloride. Okay, just not sugar, because sugar is molecular and wouldn't have conducted electricity. So that's the process you got to go through. Okay, you look at these properties and you compare them to what you have and see if you can figure out which one's which. Okay, we also said the other day that certain metals like cobalt, copper, and nickel produce certain colors. Okay, so those would be things to look for as well. Okay, it's pretty straightforward. There's only going to be a couple that will give you any trouble. You'll be able to identify like 10 of the 12 right off the bat, pretty much. Okay. All right. Then after that, you'll have your conclusion. Okay, and we said the other day that in your conclusion, you copy your hypothesis and paste it back down here, and then you explain. Okay, is this hypothesis acceptable or not based on your data? So if when this is all done, you were able to clearly identify everything, you found that all the ionic compounds conducted electricity, okay, things like that, then you can write that, okay, this is what I said in my hypothesis. Well, actually, you don't use the word me or my. Okay, this is what the hypothesis stated. The data in this experiment supports that. It said the data is exactly as was predicted. Therefore, the hypothesis is acceptable. Or if you don't find that, then you say the hypothesis is rejected because the data does not support it. Okay, and talk about the specific data. Okay. Lastly is your evaluation, 2 sources of error and ways to fix them. Right? So sources of error are anything that's not human correctable error within the experiment that could have affected the experiment. So you can't say like, oh yeah, we contaminated sample B with sample C. Okay? If you write that in the observations, this is, or sorry, in the evaluation, this is what you're telling me. Hey, Coderre, we screwed up really big. You know, we did that thing you said not to do, and then we didn't fix it. Okay, which basically says, yeah, Coderre, all of our results are meaningless because we screwed up and we didn't do anything about it. Okay, that is so not the last thing you want me to read on your lab report. Okay, a source of error is something you could do nothing about. Right? One of the things we said could be a problem was what? Exactly. We're dealing with really small sample sizes. That's a source of error. There's nothing you can do about that. I'm telling you, you can only have this much. Okay, but it could cause you to misread some things. That's a source of error. Okay, there's nothing you can do about it. Okay, and you list it. Okay, so sources of error are things that happened that could affect your results that you really couldn't do anything about in this lab. Then you can say, in the future or in another lab, we might want to do this. Okay, what's an easy way to correct small sample sizes? Have bigger sample sizes, pretty easy. Okay, you got to come up with one more. I gave you one. Okay, there's one of your 2 sources of error. All right, the class discussion questions you don't have to do. Okay, when I mark your lab and return it to you, then I'll talk about these. Okay, all right, any questions there? All right, so what you're going to spend the bulk of the rest of the class doing here now is get together with a group. Let's say like groups of 2 or 3 will be lots. Okay, make sure everybody has a group. If you don't have a group, let me know. Okay, and then you guys can sit down and start working out how the procedure is going to work, or divvy it up, and then kind of share it with each other. If your procedures are similar, that's fine. You're working on that together. But everybody's report should be at least somewhat unique. Okay, your hypothesis should be yours, not. I shouldn't see 5 the same. Okay, same with the other stuff. Okay, your analysis, all that other stuff should be unique to your lab report. The stuff you're working on with your group mates, that's going to be similar. That's fine. Okay, what I don't want to see is 5 carbon copies of the same lab report handed in from 5 different people. All right, because then I don't know who really did all the work. Okay. All right, so get together with some other people and you can get to work on the procedure part. Okay, remember, really detailed is what we're looking for here.